Here goes some more questions on 9.4. Again, everything to do with flowers and germination and seed dispersal and dicots and monocots, all of this sort of stuff, okay? Everything on 9.4. So let's just get started. The following is a diagram of a flower. Okay, so here we can see the flower and we can see there's some labels, X, Y, and Z. Now the question is obviously going to ask you what is X, what is Y, and what is Z. Okay, so let's try this by ourselves. And maybe if you're a bit confused about what's going on here, I can quickly show you something. So what they're really showing you here, okay, if we look at a flower, let's say this rose. Maybe you haven't tried this, but if you open up the flower, so you remove these petals, eventually if you remove enough of them, you'll be able to see this, okay, the little like organ sort of a plant, right? And a flower has a female, so this is the female part, and all these little male parts, okay, these yellowy ones, okay? So basically a flower can be both male and female at the same time. So that's what we're looking at, okay? Now, let's go back to the question. X. So X is pointing at this center thing, okay? We know this center thing is the female organ, okay? And it's pointing specifically at the top, okay? We call this the stigma, the sticky stigma, because it is gonna catch what the male part makes. We know the male part, all of these little ones here, they make pollen, and pollen will get transferred to the stigma by a bee or wind or something like that, okay? And once it lands here on the sticky stigma, it will get taken down here to the female or to the female cells, and basically that's fertilization, okay? So X is the sticky stigma, okay? So it's probably the answer is going to lead us to A or C, because B is saying anther and D is saying anther, and that is not the X, okay? The anther is this part here, the male part. Anyways, so we know it's going to be A or C. Now, what is Y? Y is pointing at, let me go to my picture a bit clearer, pointing at the filament holding this anther. So this big yellow thing is the anther, and this orangey pipe is called the filament. Okay, so Y is a filament. So we know automatically, without even needing to do Z, the answer is C. Okay, we already know that. But we can confirm and check if Z is really the ovary, which it is, okay, the ovary, okay, pointing at the ovary. So the answer is going to be C. Next one. So this finally, we haven't gotten to any questions yet about germination, but here we have one. What steps occur in germination after water uptake? Okay, so first of all, what again is germination? So germination is the process by which a seed becomes a plant. Okay, so here we have a seed, okay, under the soil. So this is soil. We got the seed under soil and we opened it up, okay? So we slice basically the seed open to see what's going on the inside. So what happens in germination is this. We know it's super important to give a seed water, right? Why? Because water, if we give it water, water will get taken up by the seed, okay? It will enter the seed. And it will specifically, let me move this a little bit, activate a molecule called gibberellin, okay? So we got water and it's gonna activate gibberellin, okay? We're going to say it's the soldier. And the soldier, we know, they command people. So the soldier is going to command another molecule to be made, which is this ratatouille, okay? It's like a chef. It's an amylase, okay? We can think of it, wait, where is it? Oh no, sorry, I messed it up. That's the amylase and that's gibberellin. Okay, so the soldier or gibberellin is going to wake up the amylase. And amylase is like a chef. It breaks down food and prepares food, okay? So basically amylase is going to break down maltose. Maltose is, let me show you. It's not going to break down maltose, sorry. It's going to break down starch into maltose, okay? So we got starch here, and it's going to break it down. Let me show this clearly. It's going to break starch down into maltose, okay? And basically this is important. I hope this is clear enough. Starch is broken down into maltose, because starch is too big to be used for energy, but maltose is pretty small. So if maltose is formed, it will then turn into glucose, and we know that this is small enough for the cells inside the seed to use to be able to create a plant, okay? So after a while, we'll create a plant. The plant will grow bigger. The plant will start forming and grow bigger and bigger, okay? Basically, that's what's gonna happen. That's germination. So, is it A, gibberellin is produced followed by amylase activation? This seems true because after water uptake, we make gibberellin, remember the little soldier guy, and that activates amylase. So, gibberellin is produced followed by amylase activation. So we can pretty much know it's going to be A already, but let's confirm what's going on with the B, C, and D. B is gibberellin stimulates photosynthesis to begin. No, it doesn't. It activates amylase. Okay, C. Amylase breaks down starch to glucose, which activates the embryo. So this is not what happens directly after water uptake. First, it's gibberellin. So that's why C cannot be true, because amylase is a bit later. So immediately it'll be gibberellin, then amylase, okay? And also amylase breaks starch into maltose, not necessarily glucose, okay? Now D, amylase synthesis followed by activation of gibberellin. See, that's the wrong way around. It's gibberellin synthesis followed by amylase activation, okay? So we know it's going to be what? A. Okay, cool. Next one. So here we go back to some dicot questions, okay? Monocot, dicot. What is a characteristic of dicotyledonous plants? So remember, in general, as I said many times, plants are in general grouped into either monocotyledons or dicotyledons. And they have various differences, including their flowering, their leaf veins, their roots, and some other things, okay? So here's a little table that summarizes the key differences, okay? These are the ones that you would need to know for IB for multiple choice questions or even some long answer questions where you need to distinguish and compare and stuff like that, right? So, coming back to the question, A, the flower parts are usually in threes or multiples of three for dicot plants. Let's see, is that true? Coming to this list, dicot plants usually flower in multiples of 4 and 5, not threes. Monocots flower in multiples of 3. So this one's wrong for sure. B, the leaves have parallel veins. No, that's not. I'm not going to show you, but basically monocots have parallel veins. Parallel meaning that they're next to each other, whereas branching veins are like the dicots. Okay, that's the kind of veins I'll show you. Okay, that's the kind of veins we see on most leaves. They come out like this, whereas for monocots, they're next to each other, they're parallel, the veins. Okay, so it's not B. C, the seeds contain a single cotyledon. Again, not true. Why? Because dicotyledon. Di means two, cotyledon means cotyledon, right? So basically they have two cotyledons, whereas monocot has one cotyledon. So single cotyledon would mean monocotyledon, not a dicotyledon. So it's not C. D, the root system has a taproot with lateral branches. Yes, that's true. If we look at the branches here of dicot, it's a taproot, so it goes down, and then a bunch of branches come off of that main root. Whereas the fibrous root, everything comes from one place, branching out, you see? So that's a key difference. So we know it's going to be D. Okay, now we have another germination question. Which process happens first during germination of a starchy seed? So coming back to this, first water gets reabsorbed, right? So it's going to be A, formation of gibberellin. Nope, that's second. Production of amylase. Nope, that's third. Absorption of water. Yes, that's first. D, conversion of starch into monosaccharide. That's last, okay? So it's going to be C. Final question here. What is a role of gibberellin produced in the cotyledon of a dicotyledonous seed? Okay, so gibberellin, as we show here, is made in the cotyledon. The cotyledon is here, okay? It's around this location of a seed. Okay, and that's where it's made. Now, what's its role? To open the micropyle to allow water absorption? No, right? Because water gets absorbed first, then gibberellin gets produced. Not gibberellin gets produced, and then that allows for water to be reabsorbed. So it's not A. B, to catalyze the breakdown of starch into maltose. Nope, that is amylase. Amylase does that, not gibberellin. C, to provide energy needed for growth. No. What will provide energy needed for growth is the maltose that we formed. Remember, the whole aim is to form this maltose and glucose that can provide energy, just like they do for us, okay, to make ATP. So it's not C. D, so it must be D, to stimulate production of amylase. That's true. We know our gibberellin will be made, and that will stimulate the chef, the amylase, to be produced, okay? So it's going to be D. So hopefully all these were useful for you, and again, I always emphasize this: the more you can do, the better. Email me if you want more questions. There's never enough that you can do, okay? But the more you do, you'll get into the test and you'll be like, wow, I've seen all of these before. That's because your teacher is going to use exactly these that I'm showing you, because they will also get it from past papers, okay? And the questions really, really repeat and repeat and repeat over time, if you haven't noticed yet. But so really just do as much as you can, and you'll be 100% ready for your test.